Welcome back to the channel. If you want to build a functional quantum computer today, you have to lock your qubits inside a specialized vacuum chamber, cooled to within a fraction of absolute zero. That isolation is a requirement because of decoherence. Quantum states are fragile. The heat, vibration, or even a single stray atom bumping into the system will cause those properties to instantly collapse into chaotic static. This makes the interior of a living cell a nightmare for quantum stability. As the physicist Erwin Schrödinger pointed out, the biological machinery of life is inherently warm, wet, and noisy. Yet, when we examine life at the molecular scale, we find organisms performing chemical transformations at speeds that classical physics suggests are impossible. The contradiction is stark. Either our classical models are insufficient to account for the actual behavior of biological molecules, or evolution has discovered a way to endure the exact chaotic noise that destroys our most advanced laboratory systems. We can see this in a class of enzymes called hydrogenases. These proteins facilitate the transfer of protons, and they do it at rates that far exceed the theoretical speed limits of standard chemistry. Instead of acting like solid particles that need a massive energy boost to climb over a chemical barrier, these protons engage in quantum tunneling. The particle shifts into its wavelike form, allowing it to literally leak straight through solid activation barriers to the other side. This diagram illustrates a similar anomaly in photosynthesis. Sunlight striking a leaf must travel through a molecular network to a reaction center. If the energy hopped randomly, it would lose efficiency. Instead, it forms a molecular exciton, a wave simultaneously exploring multiple pathways to its target. Traditional quantum theory suggests this wave-like behavior should collapse in the fluctuating heat of a leaf. Its survival proves that life possesses a hidden mechanism to protect its quantum coherence. For years, we assumed biology must shield these quantum states from the environment, but new research suggests the opposite. Life might actually recruit the noise to function. This waveform illustrates stochastic resonance. In this process, a chaotic background frequency doesn't bury a weak quantum signal. Instead, the signal latches on to the noise, using the ambient thermal energy to amplify itself and drive a biological reaction. However, this raises a different possibility, the faker hypothesis. What if these large biological systems aren't using real quantum mechanics at all? This theory proposes that complex biological structures, like the neural networks in your brain, might use specific mathematical architectures to mimic quantum states. By relying on immense synchronization, these classical networks could gain a quantum advantage while remaining robust in a noisy environment. Whether life is truly manipulating quantum waves or using classical networks to simulate them, evolution has already arrived at a biological solution to the decoherence problem that human engineers are still struggling to solve. It's one thing to observe a proton inside an enzyme. It's another to see how an invisible quantum quirk translates into a biological function large enough to ensure an organism's survival. We can analyze this by separating biology into hardware and software. Mammalian vision provides a clear example. Your retinal cells act as hardware, with single photon sensitivity, operating at a strictly quantum scale. Your brain then acts as the software, taking that microscopic quantum detection and amplifying it into the macroscopic conscious experience of sight. The ultimate example of this macro-quantum survival is avian navigation, specifically in birds like the European robin. A specialized protein in the bird's eye separates a pair of entangled electrons. The quantum correlation between these spins reacts to the Earth's magnetic field, allowing the bird to literally see in a magnetic map as it flies across continents. Proving these mechanisms is difficult because of a measurement trap. When we use ultra-fast laser pulses to observe a cell, we might be generating the very quantum coherence we are trying to find. To solve this, the field is moving toward the use of quantum probes. By using diamond sensors or analyzing specific photon statistics, we can observe the cell's natural state without interfering with its delicate internal balance. We are entering a stage of discovery where biology reveals itself as highly optimized quantum engineering rather than mere wet chemistry. Thanks for watching, and be sure to subscribe for more deep dives into the mechanics of our universe.